Hello and welcome to Beer Rules video lessons. Today we are going to be talking about the leaf structure. So how the leaves look like when they are seen in a cross section, they are seen inside. So just to help you visualize this is the main vein of the leaf. So this is the bottom of the leaf actually. And here we have the thickness of the leaf. So this is how thick they are. This is the thickness of the leaf. So what you're going to be seeing now is this cross section here. We are going to see how the leaf is from the top to the bottom. So let's draw it. So the first thing you're going to be having there, this is a tiny little waxy cuticle. I'm going to be showing you that later. We have a layer of cells. This is the skin of the cell, or the first layer over there. Then we have one or a few bunch of cells stacked kind of upright like this. These cells, as you can see, they have those little balls. Those little balls are chloroplasts. That means that this is a photosynthesizing tissue. These cells are going to be doing photosynthesis. They get the light and they are going to be transforming that into chemical energy. In this diagram we have only one layer of these stacked cells, but you can have two, sometimes three of them. Under that one over there, we are going to have a bunch of cells that are going to be more loosely spaced, and they are going to make like a sponge. They have air space between them. And these loosely stacked cells, they are also photosynthesizing. They also have chloroplast and chlorophyll. They can do photosynthesis, but they have another very, very important function, which is going to be related to the structure of having this air space around them. Okay. After this spongy tissue, we are going to be having, wait for it, and yes, we are going to have another kind of skin. We're going to have another epidermis, and this now is the bottom one. It has these weird looking structures over there, and of course we have another waxy cuticle there. And in the middle of this tissue we have vascular bundles. We have xylem and phloem. So this here on the top is the xylem. It has reasonably bigger cells, and in the bottom we have the phloem. How do I know which one is which? With sometimes the size is not so easy to realize of the cells. So the trick is the following. The xylem enters the leaves by the top vessels. So they go into the top vessels. They are over here. This is going to be the xylem. They are the top ones. And the production of the leaves exit the leaves by the bottom. So basically through the top comes the water and ions, and through the bottom goes away the production of the plant. So let's give name for things here. That outer muscle over there is the waxy cuticle. Then you have an upper epidermis, the upper skin. So that stack of cells over there that I show here with one stack of cells is the palisade mesophyll. Palisade is the name that is given to when you make the trenches. You put those sandbags one on top of the other, you stack sandbags. That's a palisade. So that's why this is called palisade, because it looks like a bunch of sandbags stacked, no one beside the other. Below this, we have there in the middle, in blue in this case, is the top vessel, which is the xylem. And we have the bottom vessels, which is going to be the phloem. And they are immersed into a spongy mesophyll because it looks like a sponge. Then we have the bottom epidermis, which has a stoma. The stoma are those two cells over there. Each of those two cells are called the guard cells. We are going to see the stoma in details in a few moments. Okay, just to put you together in a more weird diagram that is a little bit more clear to see, we have one, the waxy cuticle over there, two, the upper epidermis, three, the palisade mesophyll, four, the spongy mesophyll, much more loose, okay, five, the lower epidermis, six is the stoma aperture. There's an opening there. Okay, it's the middle of the stoma. You have an aperture. And seven are the guard cells. You have two cells over there labeled 7, one inside. Those are the guard cells of the stoma. And finally, 8 we have the xylem, 9 we have the phloem, and both together they make the vascular bundle. They are the tubes, the vascularization of the leaf. What about the function of the structures? We'll see in a moment. First, a few pictures for you here. The upper epidermis, palisade mesophyll, sponge mesophyll. It's not clear here if we have a vascular bundle, but here we have, look, the upper epidermis, palisade mesophyll, sponge mesophyll, and here we have xylem and phloem over there. Then the lower epidermis with two cells of guard cells making the stoma. Okay, there we have more. Okay, look at this. Upper, palisade, spongy, and there's even a vascular bundle here, not very clear which part is which because it's quite small. And now let's talk about the functions. Okay, what each part is going to be doing. The oxycuticle is going to be impermeabilization of the leaf. So basically it avoids from water or water vapor to get in, and more importantly, it avoids it from getting out. It holds the water inside of the plant. The upper epidermis is going to be the upper skin. It produces the cuticle. People love to say, oh, it protects the leaf. Yeah, okay, it protects against desiccation because it produces the waxy cuticle. Okay. 3, the palisade mesophyll is going to be the main photosynthesizing tissue. It makes most of the photosynthesis of the plant. Then you have the sponge mesophyll. What's the function of the sponge mesophyll? Exchange gases. It can allow carbon dioxide to go in and spread around the leaf, oxygen to go out and spread around the leaf and get out. So basically it's the equivalent of the lung of the cell. It spreads gases around. It also does photosynthesis, but that's a secondary function of the sponge mesophyll. The lower epidermis, it also produces waxy cuticle, so it also impermeabilizes, but it contains most stoma. And what then is the function of the stoma? The stoma has an aperture, has an opening, which allows the gases to get in and out. The plant loses oxygen and water vapor through the stoma. The plant gains carbon dioxide, especially through the stoma. So the plants are going to be exchanging things through the stoma. The stoma aperture, therefore, allows that to happen, and the guard cells are going to be opening and closing the stoma. So the guard cells, they are going to be very important to hold the gases inside or outside. Actually, oxygen and carbon dioxide, it's quite impossible to stop them from going in and out. There are other ways for them to go, but the water vapor is regulated by the stoma, because if the stoma aperture is open, the guard cells open and let the stoma to be open, the water vapor gets out of the cell. If the plant doesn't want to lose water vapor, then they close the stoma aperture by change the shape of the guard cells. Okay? So the xylem, you already know, it carries water and ions, is the same functions as in the stem. The phloem is the same thing as in the stem, is going to be carrying the production, glucose, amino acids, lipids, whatever. Okay? And the vascular bundle is the xylem and the phloem together. They are going to be both of the vascularization together, making the tubes that carry liquids around. Okay? So, let's take a closer look now on this stoma thing. This is a view of the surface of a leaf. We have here 1, 2, 3, 4, 5, and probably there's another one here, another one there, another one there. It's stomas. And if we look close, the stoma is going to be looking like this. You have a ring of cells of the skin that leaves an opening over there, and this opening will contain 2 guard cells. You have one guard cell here, another one over there. And these guard cells, they have the potential to be open, so they allow the opening here to let gases to be exchanged, especially water vapor, or they may be closed. So opening and closing the stoma is how the plant avoids water vapor from going in and out. Okay? Here we have the guard cells when they are turgid. Turgid means they are hard because they gained water. So if there's lots of water in the plant will push water inside the guard cells, and the natural shape of the guard cells when they are turgid, they are hard, is to have this bean shape. And when they have this bean shape over here, they let an opening. They are open. If the plant gets dry, if there's not much water, the plant loses water, so the water gets out of the guard cells over here, and the guard cells get flaccid. And if they get flaccid, they close, and now there's no way for water vapor to get out from here. Okay? And the question would be, why to lose water? So this is actually the transpiration in two different kind of plants, and we can see that in different times of the day they have different rate of transpiration. The red one, the plant A, doesn't transpire much in the night, and then at midday it transpires a lot, and then it transpires less, which is quite of makes sense because it's expected a higher temperature will be somewhere here around midday. But if we take a look in this other plant, it also has low transpiration here, a little bit high but not so much over there, and low here back again. So why the plant would want to lose water? So that is a question for another lesson, because water is very, very important for the cell. The water is what plants use to be alive and to do photosynthesis. Photosynthesis, carbon dioxide plus water to make glucose and oxygen. So why losing water? That is the question. And now let's do some exercises. If you point your cell phone camera to this QR code, it will open our quiz. You can also type this link under the QR code in your computer to be able to do the quizzes. I hope you have fun.